Structure of eukaryotic cell. Plasma membrane. Plasma membrane is a dynamic fluid structure and forms the external boundary of cells. It acts as a selectively permeable membrane and regulates the molecular traffic across the boundary. The plasma membrane exhibits selective permeability, that is, it allows some solutes to cross it more easily than others. Different models were proposed to explain the structure and composition of plasma membranes. In 1972, Jonathan Singer and Garth Nicholson proposed fluid mosaic model, which is now the most accepted model. In this model, membranes are viewed as quasi-fluid structures in which proteins are inserted into lipid bilayers. It describes both the mosaic arrangement of proteins embedded throughout the lipid bilayer as well as the fluid movement of lipids and proteins alike. Chemical constituents of plasma membrane. All plasma membranes, regardless of source, contain proteins as well as lipids. The ratio of proteins to lipids varies enormously depends on cell type. Carbohydrates are also present in plasma membranes and comprise only 5 to 10% of membrane mass. Carbohydrates bound either to proteins as constituents of glycoproteins or to lipids as constituents of glycolipids. Carbohydrates are especially abundant in the plasma membrane of eukaryotic cell. Lipid bilayer. The basic structure of the plasma membrane is the lipid bilayer. This bilayer is composed of two leaflets of amphipathic lipid molecules whose polar head groups are in contact with the intra or extracellular aqueous phase, whereas their nonpolar tails face each other, constituting the hydrophobic interior of the membrane. The primary physical forces for organizing lipid bilayers are hydrophobic interactions. Three classes of lipid molecules present in lipid bilayer are phospholipids, glycolipids, and sterol. Other than these, lipids are the special constituent present in the plasma membrane individually. Phospholipids. Phospholipids are made up of four components: an alcohol, which is glycerol or sphingosine, fatty acids, phosphates, and an alcohol attached to the phosphate. The fatty acid components are hydrophobic, whereas the remainder of the molecule has hydrophilic. There are two types of phospholipids: glycerophospholipids and sphingospholipids. Phospholipids derived from glycerol are called glycerophospholipids. Glycerophospholipids are also known as phosphoglycerides, contains glycerol, fatty acids, phosphate, and an alcohol, which is example choline. Phosphoglyceride molecules are classified according to the types of alcohol linked to the phosphate group. For example, if the alcohol is choline, the molecule is called phosphatidylcholine, which is also referred to as lecithin. And if serine, then it is called phosphatidylserine. Phosphoglycerides are the most numerous phospholipid molecules found in plasma membranes. Sphingospholipids. They contains an amino alcohol called sphingosine instead of glycerol, a fatty acid, phosphate, and an alcohol attached to the phosphate. In sphingophospholipid, the amino group of the sphingosine backbone is linked to a fatty acid by an amide bond. Sphingomyelin is the most abundant sphingophospholipid. The plasma membrane of animal cells contains four major phospholipids. The four major phospholipids are phosphatidylcholine, the most abundant glycerophospholipids in the plasma membrane, phosphatidylserine, phosphatidylethanolamine, and sphingomyelin. At neutral pH, the polar head group may have no net charge, is in phosphatidylcholine and phosphatidylethanolamine, or it may have net negative charges like phosphatidylinositol and the phosphatidylserine. Rarer phospholipids have a net positive charge. Glycolipids. Glycolipids contain carbohydrate, either monosaccharide or oligosaccharide, covalently attached to the lipid. These can derive from either glycerol or sphingosine. The simplest glycolipid, called a cerebroside, contains a single sugar residue, either glucose or galactose. Gangliosides are most complex like glycolipids containing a branched chain of as many as 7 sugar residues. The glycolipids are found exclusively in the outer leaflet of the plasma membrane with their carbohydrate portions exposed on the cell surface. Sterols. The basic structure of sterol is a steroid nucleus consisting of 4 fused rings, 3 with 6 carbons and 1 with 5. It is planar and relatively a rigid structure. Cholesterol is the major sterol present in the plasma membrane of animal cells. The plasma membrane of plant cells lack cholesterol, but they contain other sterols like stigmasterol and cytosterol. With rare exceptions like mycoplasma, bacterial plasma membrane also lacks cholesterol. Lipids. Lipids are not randomly mixed in each leaflet of the bilayer. Certain lipids in the plasma membrane, particularly cholesterol and sphingolipids, are organized into aggregates called lipid rafts. Lipid rafts are membrane microdomains that are enriched with cholesterol and glycosphingolipids. These microdomains also contain specific proteins. The rafts in cells appear to be heterogeneous both in terms of their protein and lipid content and can be localized to different regions of the cell. There are two types of lipid rafts, planar, which are also called as non-caveolar, and a caveolae. Caveolae are flask-shaped invaginations of the plasma membrane that contain caveolin proteins. Caveolin is an intrinsic membrane proteins, is a cholesterol binding protein. Lipid rafts have been implicated in processes as diverse as signal transduction, endocytosis, and cholesterol trafficking. When amphipathic lipids are mixed with water, three types of lipid aggregates can form. In the case of fatty acid salts, which contains only one fatty acid chain, the molecules form a small and spherical micellar structure of diameter usually less than 20 nanometer, in which the hydrophobic fatty acid chains are hidden inside the micelle. The second type of lipid aggregate in water is the bilayer, in which two lipid monolayer combine to form a two-dimensional sheet. In the third type of lipid aggregate, lipid bilayer forms a hollow sphere called a liposome. Liposomes are closed, self-sealing, solvent-filled vesicles that are bound by only a single bilayer.